presenting is Nexsen Proprietary Limited. Nexsen is an Australian diagnostic company focused on building a point-of-care testing platform, utilising proprietary biomarker discovery and nanoparticle engineering to develop material improvements in testing outcomes. To tell you more, I am joined by CEO Thomas Hanley. Good morning, Thomas.
Good morning, Jane. How are you? And thank you for everyone joining this call.
No worries. I'll just get you to pull up your presentation and jump into it.
Cool. First of all, thank you very much for joining the call. My name is Tom Hanley. As pointed out before, I'm the Managing Director and CEO of a company called Nexsen Limited, a company that was formed back in 2022 expressly to develop and design and commercialize a range of diagnostic tools or diagnostic applications for various pathologies and diseases that are quite common around the world. So I'll go right into the presentation. The basis of the technology for Nexsen is the development with our team at RMIT University around biomarkers, which we refer to as aptamer biomarkers. Aptamer biomarkers are those that are short-chain biomarkers and are distinguished from the current biomarkers that are used in a lot of diagnostic testing, which are called antibodies. So antibodies are a live, long-chain DNA sequence, whereas an aptamer is a short-chain DNA sequence and synthetic, which enables us to develop and test and release these in a lab environment and into commercial space in a much shorter period of time. Around the key sort of technology of aptamers are the two basic tests or platform tests that we've designed. One sort of in that more sort of simpler clinical environment, which you see on the left-hand side, which looks a little bit like a rat test, but has a lot sort of more smarts going on. And on the right-hand side, we have what we call the cylindrical test. And both of these combined provide us with what we call the lab on a stick experience. So a lot of sort of diagnostics at the moment and sort of diagnostic development are looking at improving diagnostics either in a PCR environment or in a lab scale pathology-based environment. And what Nexsen is doing is developing a series of diagnostic tests that we call lab on a stick, which means instead of having to go through the expensive and timely process of developing or taking a test in a lab, and then having that test cultured and the results coming back within sort of 1 or 2 days. These are rapid COVID-like tests that provide a result in a very, very short period of time. So excited was the government around the sort of work that we're doing with our first test that we were awarded the CRC-P grant. A grant that provided us up to $3 million in cash, matching funding and in-kind contributions from RMIT and Northern Health, which I'll explain a little bit later. This is just a shot here showing our lab at RMIT and the developing of our first diagnostic point-of-care test, which we call StrepSure, and I'll come to that very shortly. So what we've achieved to date is we've signed a commercialization and research agreement with RMIT. We've completed the lab-based testing for our first test, which is the test around Group B Strep. So Group B Strep is a very, very, I guess, difficult pathogen. It is a pathogen that, or a bacteria that passes from mother to baby during delivery. And the world is kind of made up of a number of different ways of dealing with Group B Strep. In Australia, we have testing that is done on women done at 36 weeks in a lab-based environment. Many other countries follow that. We've got the United States, we've got Europe, Japan, and a couple of others. But mostly countries do not test or have very sort of ad hoc approaches to testing for Group B Strep with pregnant women. And the result of that is that when Group B Strep passes from mother to baby during delivery, baby is then exposed to things like sepsis and meningitis, which means that Group B Strep is one of the leading causes of neonatal deaths and neonatal disabilities globally. So we've completed the lab-based testing. We were awarded the Australian Commonwealth grant of 7.7 million, which includes 3.5 million of cash. We have selected and had approved our clinical trial partner, of which we've made ethics approval for our first set of clinical trials. And we've done the design and development through our technology partner, Design Industry. Further to that, we were then awarded a second grant. Normally, for a company our size, getting one grant of this nature and this magnitude is incredible. We then received a second grant, an ARC or an Australian Research Council grant, for our development of a mastitis-based test for cattle. We've also identified other clinical marketing opportunities, launched our GBS Aware platform, and have in the process of developing what we call Nexsen.ai, which is an AI platform, which I'll come into shortly. So as mentioned before, these are our partners, RMIT University, who has been diligently providing us with the technical research around the aptamer development and the biomarker discovery for things like Group B Strep. They're also in the process now of looking at biomarker discoveries for other things like, for example, kidney function. So around kidney function, we're looking at a rapid test that measures creatinine levels in the blood, as well as levels of NGAL, which gives us a sort of a perfect rapid point of care kidney function test, ideal for a lot of patients who suffer in that sort of type 1, type 2 diabetes or have other kidney-related disease issues. Again, partnered with the Australian government around the funding that we received both from Department of Industry and Science and the Australian Research Council, and our clinical trials through Northern Health is the ethics approval is now well underway. So overview of test one as sort of Group B Strep. When we were awarded the grant to develop the trial or the rapid diagnostic test for Group B Strep, it was stressed that even in countries like Australia, where women present at 36 weeks for their Group B Strep test, there are still women who either miss that test or, because Group B Strep is such a transitional bacteria, they can still contract Group B Strep between 36 weeks and delivery. So the ideal test is a test that can be done at hospital as she presents for delivery. And once they know that she's got Group B Strep, the ability to then give her the antibiotics, which is just simple penicillin, means that that baby will not be born with the possible sepsis or meningitis. Now, that's in the Australian context. Globally, having a cheap, inexpensive, rapid point of care test will address the 134 million births given globally, of which very, very few are ever tested for Group B Strep. And I wanted to stress with this particular slide, I guess, the global impact of something like Group B Strep. So Group B Strep is not something that is just a poor disease or a rich disease, a tropical disease, or a disease that affects one particular population. Group B Strep affects every country in the world. And unfortunately, many of them don't deal with Group B Strep, particularly in developing countries. And as a result, there's where you're getting your high cases of sepsis and meningitis, your deaths, your long-term disabilities. When we were awarded this grant, it was quite clear, and I think it kind of goes back to maybe one of the previous presenters' slide deck, is that Group B Strep leading to sepsis, meningitis, deprivation of oxygen to the brain, long-term disability, a massive impact to the NDIS. And as a result of that impact to the NDIS, these are costs that are not just a one-off but ongoing in terms of supporting that child. In the UK, for example, there are no universal screening options for Group B Strep, and as a result, the legal fraternity has stepped in and is providing legal support for families to sue the NHS and the UK government just for the funding to support children with long-term disabilities as a result of Group B Strep. So that was really our first sort of really important test, and the one that we're taking to market in the first step. As I mentioned before, we then won another major grant with the Australian Research Council to take, because Group B Strep is one of the factors in mastitis in cattle. Mastitis is one of the largest impacts or financial impacts on the dairy industry globally. To have a rapid test to detect if a cow has the GBS bacteria or the 2 or 3 other conditions that are required for mastitis, and to be able to isolate that cow will save farmers globally billions of dollars a year as a result. And again, one the government was quite happy to support because of the impact that it has to the Australian dairy sector, as well as others around the world. Execution roadmap. So we have completed our last round of funding. We have acquired the patent for the GBS antibodies from RMIT. We've completed or about to complete the final prototype. I'm actually in Singapore at the moment to discuss manufacturing options with 2 or 3 companies over here. We've made our submission for ethics approval for our first trial, and we are in the process, and we have signed off and started the aptamer discovery for creatinine and NGAL for our kidney function test. Again, just want to sort of highlight that while the sort of aptamer discovery and having the ability to have these very, very important biomarkers, which enable us to detect the particular pathogen or disease as accurate, right, as you would get in a lab-based environment, not only requires the chemical intervention that we're working on, but also a device that is able to be used both in a hospital setting or in a home setting, whether it's sold as a retail product or as a clinical product. We want something that sort of does away with the kind of rat test COVID test scenario where you're sort of fumbling around with little vials of liquid and sort of contaminating the whole process as you go through. So one of the design one of the platform products is what we call a cylindrical test, and so there's up to 7 processes that occur inside that test, to the point where then the liquid then drops down onto the lateral flow device, and at that point we're getting the kind of sensitivity and selectivity that we would be getting if it was done in a pathology test. And we're already getting those results in the lab-based tests that we're doing at the moment. So the lab data strongly supports that the work that we're doing in this aptamer discovery, or this biomarker discovery, in the development and improvement of selectivity and sensitivity, are giving us the results that you would get in the lab. And the really important thing here is that, as we go through and look at the future tests, this is about making diagnostics more available, easily scalable to everybody. Now, obviously, on the left there, we have the kidney function test. This is a test that impacts people with type 1, type 2 diabetes. It is a type of test that is done virtually every time you go to hospital in ER. They will do a creatinine or kidney function test. So if you ask many clinicians, the kidney function test is probably the second most requested pathology or diagnostic test outside of a glucose test. And at the moment, someone suffering from a comorbidity like, say, type 1 or type 2 diabetes would have to go into a clinic and get that done. Whereas now they'll be able to buy this or have this and do these tests at home without having to go into a clinic to do so. One of the other tests, which is near and dear to my heart, the rapid point of care test for PSA, or prostate specific antigen. As a sufferer of prostate cancer myself, I am stunned, or was stunned, at the lack of interest from other friends of mine who just aren't interested in getting a PSA screening test. And it was revealed to us that the PSA test market, particularly the one that you're able to then take at home in that sort of privacy, is something that is quite significant. So that sort of rounds out the sort of first 4 tests that the company will be releasing. Mentioned before AI. So one of the key elements here in the development of aptamers is working with a machine-based algorithm that will help us identify and optimize and provide a series of aptamer sequences prior to then going in and conducting further tests in the lab. So the idea behind the platform here is to build a system that would reduce the time of discovery and optimization. And we're looking probably upwards of 3 to 6 months of time reduced to get a new aptamer or biomarker tested prior to clinical trials. This is our leadership team. So myself, Tom Hanley, Mark Muzzin. Mark has worked on several biotech companies and other ASX-listed companies, and is a strong liaison with us with the Australian Research Council and RMIT University. Gavin brings significant business experience and commercialization experience through his 30 years, as well as being on two other ASX-listed companies, and Martina's expertise in regulatory and other medical related things that we have to follow through on is second to none. So this is the board team here. I know that Jane likes us to keep us moving quickly, so I'll finish the presentation there and take questions.
Wonderful. Thank you so much for that. And again, if you do have questions for Tom, please use the Q&A function, which can be found at the bottom of your screen. Tom, I might actually just get you to stop sharing there. And we do have quite a few questions just about the aptamers here. So perhaps if I'm interpreting this correctly, can you give a little bit more insight as to why aptamers have an advantage over traditional diagnostics that focus on antibodies?
Yeah, so antibodies take a lot longer to develop. In many cases they're because they're a long chain DNA sequence and they come sort of from a living perspective. The time in the lab, the time to optimize, because it's such a complicated sequence, is a lot longer than, say, an aptamer. So the time to discovery and the time to optimize are greatly shortened when you're working with aptamers over antibodies. You're not requiring live subjects like animals or anything like that to help culture. And the shelf life, because it's a synthetic-based sequence, is a lot longer in an aptamer than it is in an antibody. So the ability for us to distribute these globally, to look in areas where, say, you've got, because in a lot of cases with diagnostics, you're looking, or other sort of medical devices, they strongly recommend you have to store them between a certain temperature and a certain conditions in order for them not to expire. We get a greater range of those temperatures and conditions when using an aptamer over an antibody.
Wonderful. And again, there's quite a few questions here and just some interest in the Nexsen AI component of the presentation. So I'll summarize them and just ask about the competitive advantages compared to traditional methods, and perhaps what kind of improvements to efficiency and findings are you seeing thanks to what we're doing at Nexsen AI rather?
Yeah, look, so very, very good question. So I just want to stress that we're obviously not replacing the lab-based process here. What we're doing is we're trying to create the opportunity to reduce the time of discovery and optimization and provide the researchers with a range of aptamers and that then they can then go through a lab-based process. So we are at the very sort of beginning of the Nexsen AI experience. We've signed off a project with RMIT. We're in the process of signing off another part of this using the deep learning supercomputers at CSIRO. But we see over the next sort of 24 months, this really sort of reducing that discovery time by 6 months, by then having access, first of all, to the experience that we've undertaken into discovery these first set of aptamers for GBS, but also being able to access the genetic databases that are available globally that will then have us look at things like PSA, prostate-specific antigens, to look at other types of cancers, and other things like Strep A and other diseases that we could have a look at as well. So all these sort of DNA sequences are available, and it's about developing the right tool to then be able to look at creating the right biomarker, the right binding to those particular pathogens for our aptamers.
Thank you, Tom. And just final one, and this one's come through again a few times. It's just on commercialization. So do you want to just perhaps give a little bit of background on how the company plans to commercialize the product?
Sure. So as I mentioned before, and I just weary of your time there, Jane, but just as I mentioned before, so when we started off this project, we funded it, myself, Gavin, and a couple other investors backed this project with RMIT because we saw this first test in Group B Strep. My wife's a midwife. We realized the importance of how Group B Strep sort of forms part of our first test equation. The funding, obviously, from government will basically cover the cost of getting that product to a point where it's ready to commercialize. So that's really a big part of the spend has been taken care of from government. The hospitals that we're working with have already indicated that as long as the sensitivity and selectivity is as good as the current testing regime that's out there, then they will switch over to using our tests. So that's the feedback that we've had. So in the process of commercialization, we've got the clinical trial. We're going through patient recruitment. The wonderful thing is that working with Northern Health, which is the largest hospital group in Australia, they are part of our grant process. So they're partners with us in this grant. They're co-contributing to the funding and their contribution to the development of this project through their contribution into the clinical trial. That will take around 6 months. And then once that's completed, along with the ISO 13485 that we're developing at the moment, then we will start making our applications to FDA and TGA for those. And then we'll go into that sort of commercialization phase of sales. But the indication has been so strong that hospitals have asked to be part of this clinical trial, of which unfortunately we've had to say that we're doing, because we're doing a sort of 1 or 2 site clinical trial, that we've kind of had to push away a number of hospitals around that because the interest is so strong, because it's such an important test. And because, and just to leave this, the test is one part of it, but the treatment is just simple antibiotics. So the ability to have that test done at that right moment, and for the result to begin as antibiotics for that child not to have sepsis or meningitis, is quite a powerful statement around what it is that we're trying to do. So,
look, wonderful. We have run out of time here, but again, copies of recordings of today's presentations will be available online in the coming days. But Tom, exciting times, and thanks for joining me.
Thank you very much, Jane, and thank you very much to the audience.